So, welcome to week 3 of uh, NPTEL Computational Systems Biology. So, last week we discussed about one thing. Nobody has joined yet, but still I'm starting because it's past 6. I have to start. So, uh, I think people will join in due course. Let's get the class started. Okay, so welcome to week 3. Last week we discussed about network properties. Graph properties. What are the different types of parameters by which we use to describe graphs, different types of degree distributions, and everything we talked about. This class will take it a little bit further. We'll learn about certain graph algorithms, we, specifically how we can do partitioning of graphs using different algorithms. And other than that, we'll give a few examples about how uh, this concept of networks and different parameters of networks can be used to study network biology as well. So let's get started. Okay, so the first concept here today is assortativity. So, intuition-wise, what assortativity means is actually a similar type of nodes, similar functional nodes will be connected to similar nodes of similar function. So, for example, if you say about proteins of similar function will be connected to proteins of other proteins of similar function. In social networks, you can talk about people having a particular uh, interest will be connected more with people of having that particular interest. So the concept is simple, that you have similar type of nodes that actually connect together. So this is true across a lot of uh, network uh, network examples, including biology. So in our case, in biological cases, we are more concerned about network assortativity, where basically the same type of genes or proteins that are connected to each other, and how we can use this concept to study a particular phenotype or a disease, etc. So degrees are a metric that is measured on individual nodes. So we quantify the degrees of a particular node. We can extrapolate that to a network by averaging out the number of degrees of all the nodes in the network, and also we can use degree distribution as a proxy to that as well. But generally, this is calculated on individual nodes. But when we focus on the edges, there are always 2 degrees associated with each edge, which is one for the node that from which the edge originates, and from the node that it actually points to. So this is more prevalent in directed networks. So there will be as we talked about last week, there's an in degree and an out degree. So in degree is basically the number of uh, edges that are coming into the node, and out degree is the number of edges that are going from the node to other nodes. But here we are talking more about the edges, from which node does the edge originate and to which node does the edge actually point to. So if we kind of take these values and plot a scatter plot, we can produce a plot that actually gives us the information about possible degree correlation between nodes across the edges. So such correlations of node properties across edges can be generally described as the concept of assortativity, which meaning that the nodes of similar function will generally tend to connect with each other. So yeah, that is how it is defined. The tendency for assortativity, which is like positive assortativity, is the tendency for the nodes to connect to other nodes with similar properties within a network. And assortativity is actually the opposite of that. The formula is given by this parameter, which is R is the assortative index of a particular node, and basically yeah, so this is the formula where E is a 2 by 2 matrix, which indicates the fraction of edges between nodes of different types, and each element inside the matrix represents the fraction of edges in the network between a node of type I and a node of type J. And the trace of the matrix is actually the sum of the main diagonal elements. That is how a trace is defined. And basically, this equation is basically will give you an index. Higher the index, this is like a correlation coefficient. So higher the coefficient, the more assortative the network is. Similar nodes connecting to each other. The lower the index, the more is the negative assortativity or the assortativity. Okay. So this is just an understanding of certain properties of a network that we'll use to study networks, right? Assortativity is one of them. Now, very similar to this concept is the centrality lethality hypothesis. So, as I was talking about, this assortativity means that similar nodes of similar function will be connected to each other. And what the centrality lethality hypothesis predicts is that uh, nodes. I've written here proteins because we are more concerned with the biological elements rather than the other kind of networks. But in general, the nodes with a high degree centrality are likely to be more lethal. So, for example, assume a protein-protein interaction network. So, you have a whole network inside a cell. You have created a network inside the cell, which actually connects all the proteins that are inside the cell and how each of them modulate each other. Now, if you imagine a protein that is actually connected to a lot of other proteins, that particular node or that particular protein has the highest number of degrees that are associated with it. So what it means is it is very essential to the network. So without that particular protein or that particular node, it's very hard for the network to sustain itself. So the centrality lethality hypothesis iterates that only, that higher the centrality, more is lethal is that particular protein. So, for example, for intuition, the proteins involved in more interactions are likely to cause death when knocked off. So, those actually become targets for different drugs. So, for example, in a cancer cell, you construct a protein-protein network, and you find a protein that is highly connected, most connected than other proteins. So, the idea would be to knock that protein off so that the cell can die. It's highly likely if you knock that off, the cell will die. So these kind of applications is there for this kind of hypothesis, right? So yeah, this proposition gave rise to different several new investigations in which stronger associations were obtained for other centrality measures. Uh, most of the protein-protein interaction network earlier used to focus on networks of S cerevisiae, which is basically yeast. And in recent study, it was found that degree and betweenness of lethal proteins is significantly above average across 20 different protein-protein networks. Closeness centrality, on the other hand, did not perform as well. So betweenness and degrees we have talked about in the last class. So betweenness is basically uh, telling you that how many paths does that particular node connect to. So basically you have other nodes in the network and you are wanting to measure the betweenness centrality of that particular node. What you need to like know is basically how many of the other node connections, other two nodes in the network, how many of those connections are through this particular node. So higher the number, higher the betweenness centrality, more essential that node is for that particular network. It is quite intuitive if you think about it. So uh, yeah, so higher the betweenness centrality, higher is the propensity that this particular node is very important. And if you knock this node off, it's most probably the network will not be able to sustain itself. So that is what the centrality lethality hypothesis tells us. Now, there's an index to measure how crucial an individual element is uh, is is for a particular network to sustain itself, and that is what is called as the pairwise disconnectivity index. So it is basically an index that quantifies how crucial an individual element is for sustaining the communication ability between connected pair of vertices in a network that is displayed as a directed graph. Such an element might be a vertex, it can be an edge, as well as a group of vertices and edges. That is, index can be viewed as a topological redundancy, a measure of topological redundancy of regulatory paths which connect different parts of the network as a measure of robustness of the network. Remember, we talked about robustness is basically how capable is the system uh, to come back to its original state after being perturbed. That is what is robustness is defined as. So basically, if you, in context of protein-protein networks or gene regulatory networks, how does what happens when you knock out a gene or knock out a protein inside that network? How well that network actually is able to overcome that perturbation. So that is what is measured. But in this case, this pairwise disconnectivity index can be a measure of the vertices and as well as the edges as well. So nodes and edges we both are considering for this particular index, and it can be a group of nodes and edges as well. And it gives you how much robust that particular network is to and how important is the presence of that particular element to the network to sustain itself. Right. So yeah, this is the expression by which dissociativity is measured. So here your n naught is basically the number of vertex pairs in the network that are connected by a path of any length in the network, and NV is the number of vertex pairs that remain connected following the removal of V. So this is basically a fraction which will give you. So basically N naught is the total number of vertex pair connections. So for example, you have 10 nodes and there are 10 or 20 edges connecting it. So there is 10 pairs of vertex pair connections between all the nodes in the network. Now you remove one node and then you measure how many of those connections actually remain. So, and you divide it by the original number. So this gives you a fraction. Higher the fraction, more is your basically more robust is your network. So higher the fraction, what it means is basically if your NV is sorry, lower the fraction, the more connectivity connected is your network. Sorry, I did not understand. Slide as in? Okay. I hope you can see my screen, right? Is there an issue with seeing the PPT? Uh, okay, so when I'm doing this, like, can you see the whole screen, or is it like you see it still in non-presentation mode? Because I am showing this in the presentation mode. Yeah, I hope you can see my screen. Okay, good. Please let me know when you cannot see the screen. Okay, so technically, this value will be 1 if your n minus v is basically 0. So if your n minus v is equal to 0, what it means is after you remove that particular node or a set of vertices or whatever you're removing, none of the connections actually remain. So disconnectivity index of 1 means that particular node or that particular set of nodes or vertices or whatever you're removing is actually very essential to the network. If you remove that, none of the rest of the network actually remains. So higher the number, more important is that particular element to the network, right? So ideally, you should, if you have a very robust network, you should be able to bypass removal of a few nodes and vertices, and your disconnectivity index should be lower. Right? It's a very simple intuition to understand. Okay, so this is an example of actually a protein-protein interaction network in yeast. So the largest cluster inside this particular protein-protein interaction network is contains almost 78% of the proteins shown. And the color of the node signifies the phenotypic effect after removing the corresponding protein. So for red, you have the red means actually those are lethal. If you remove those nodes, the phenotype is lethal. Green shows non-lethal, and orange shows slow growth, and yellow is unknown. If you notice, most of the reds are concentrated actually within the center of the network, and less on the periphery. You have peripheral red nodes, but most of them actually are concentrated inside the center, which means more connected the particular node is, the more lethal it can become when you remove that. And this is actually a connectivity distribution showing the uh, interacting yeast proteins, giving the probability that a given protein interacts with k other proteins. The exponential cutoff 6 indicates that the number of proteins with more than 20 interactions is slightly less than expected for pure scale free networks. In the absence of data on linked directions, all interactions have been considered as bidirectional. So this is actually a log plot which shows you that once you cross a certain threshold of interactions uh, for particular proteins, it is slightly less expected that is happening inside a particular network. So, for example, what it means to say is, in most networks, in most real life networks, or scale free networks are real life networks. So, in real life networks, what you'll see is you'll have very few of these proteins or nodes or edges which will have very high disconnectivity index as I defined here. So if you remove what it means is, if there will be very few nodes inside the network which when perturbed will completely collapse the network. So that is how real life networks are made. So you'll have certain nodes inside the network which will be lethal, but the number of certain such nodes will be lower. So once you so here what it's showing is, if you have more than 20 interactions, it is highly probable that nodes like those do not exist inside this particular network. And this is actually a real life network of yeast for protein-protein interaction networks. And so this is actually the third plot, which is the C, is actually showing the fraction of essential proteins with exactly K links versus their connectivity K in the yeast proteome. And this is basically the number of links, and this is the number of essential proteins. So if you see, more the number of links, more percentage of essential proteins are connected like that. So the more the essential protein, the more the protein is essential, the more connections it will have. This is what is shown here, and this is basically shown on a real life network, so that like it serves as a good example, and to example to show to. Right. So yeah. Okay. Now, we've talked about different type of parameters by which you can measure how lethal a particular removing a particular part of the network can be. And now we'll talk about how this network perturbations actually work in biological networks. Sorry. So, interactions among molecules, cells, and individuals determine the properties and the behavior of the biological system. These interactions give rise to various biological networks and their mathematical representation as graphs. We can use biological networks for integrating different data types. So, as we have talked about in earlier classes, we can have different types of these networks. You can have these gene regulatory networks, you can have protein-protein interaction networks, you can have gene-protein networks, you can have gene-disease networks, whatever. So, there's a lot of combinations possible, and each of these networks can be used to understand how actually what is the actual mechanism of working of for a particular phenotype to appear or a particular gene to express, etc. Right. So molecular changes in many cases, which will be mutations, can be regarded as perturbations. So in our case, if you assume a gene-gene interaction network or a gene-protein gene disease interaction network, Mutations will be what that causes these perturbations. So, say for example, you take this particular network. This is a transcription factor that is connected to these three other genes, and now if there is one mutation here, actually this connection gets lost because if a transcription factor is mutated at the position where it goes and binds to the gene, that particular transcription factor will not be able to go to the gene and bind, and therefore not be able to express that particular gene. So in these kind of cases, these perturbations can be viewed as mutations, right? Mutations which do not allow the connections between these two set of elements, the transcription factor and the gene. Right? And the
Hi,
yeah,
uh, can you hear me?
yeah,
So,
yeah, sure.
I think you were still on centrality lethality hypothesis slide, so I think it's not in presentation mode, I guess, or it's not changing.
Okay, okay. Just a second. I'll stop presenting and I'll present. Can you see my screen?
Yeah, I can
Can
see
you
now.
see the
Yes.
networks? These two examples.
Yes, now I can see that.
So you missed this slide.
Yes, the
Okay.
one with the east
Yeah,
connections.
yeah.
Yeah.
So basically, I'll go through it once quickly again. So this is basically a protein-protein interaction network of yeast. And this plot on the right below actually shows on the x-axis is your number of links, which is basically the degree, and on the y-axis is the percentage of essential proteins. So what it shows is higher the number of links, which is in this case 20 here. Uh, most essential proteins have higher number of links. So it follows a straight line with a positive slope. If you plot a line here, it will be a straight line with a positive slope, which means increasing the number of links means that that protein is more essential to the network or to the particular phenotype. So this is from a real world data from a scale free network. So this is a good example to uh, show basically the centrality lethality hypothesis only. So, the more connected your node is, the more essential is your node to the network. And if you perturb that particular node or the edges, whatever, it can cause lethality. So here are all the nodes represented by red are the lethal nodes, green are the non-lethal, and orange and yellow are the other parameters here. So if you can see the red ones are mostly concentrated inside, and I am not sure if it is very visible from here, and also I also cannot quite make it out, but
Yeah, I can see.
Yeah, so the idea is that your red, uh, so for example, let's take this example. This particular node is red, right? And it is connected to so many green ones. So most probably this can happen that this particular node will be lethal if you remove this, right? So
Yeah.
the nodes on the edges which have few connections will be on the greener side, and the nodes on the inside which will have more connections, they will be on the red side, which is basically lethal if you remove them. So this is an example from a yeast protein network. So yeah, that is all. Okay,
Yeah.
moving
Thank
on.
you.
Yeah. So yeah, I was showing this particular slide where basically we this is basically a gene regulatory network example. So in gene regulatory network, you have genes and transcription factors which control the expression of these particular genes, and a perturbation here can be assumed as a mutation, right? So for example, this transcription factor actually regulates three expression of three genes here. Now, if this particular transcription factor is mutated, it cannot go and bind to one particular gene, which is G1. So this part of the network will actually be redundant. So there's a mutation and it cannot go bind to this particular gene. So this part of the network becomes fully redundant, and you cannot have this particular end product of the protein or the transcription factor that is getting generated here, because from the other two genes, the products that are coming. So for example, here the second gene is forming a product which is TF3, which goes and binds to TF2, which actually regulates G5 to form TF5. Now, once TF2 is not there, it is not possible for TF3 to regulate G5 to form this transcription factor, which can do the other things that is it is supposed to do. So this is example of a healthy gene network. The moment you mutate one of these transcription factors, this actually forms to some sort of disease, because this particular transcription factor will not be generated and it will not be able to do its work, which can lead to a disease or disbalance in phenotypes, etc. So, systems like these, in systems like gene regulatory networks, this these perturbations can be imagined as mutations. It's actually the other way around. The mutations can be regarded as the perturbations of the network, and the connectivity of the network is used to predict the effect of the perturbation on the system. And you can use a host of methods to actually estimate how the perturbation is actually affecting the phenotype or affecting the network. But yeah, this is a straightforward example to say like how it might be affecting. And uh, so mutational profiles are actually come in very handy when basically predicting these kind of things. So, say for example, this is a disease network. You know, now you have been given a network, a much bigger network than this, and you have all these connections here. Now you are asked, say for example, if I perturb this part of the network, what actually gets affected really? So predictions can be done, in silico predictions can be done from these kind of networks to say that if I perturb this particular node, in reality, that node is a gene. So if you put this particular gene or transcription factor, this is what it is in downstream is going to be affected. In this case, if you affect this particular mutation, the downstream affected transcription factor will be TF5, and you can predict a phenotype or a disease that can be that can form due to loss of that particular connection in the network. Now, this can be in the other way around as well. So, for example, in a cancer cell, you are trying to kill that cell, and say, for example, you want to try to kill remove all the proteins for cell cycle inside the cell, and so that the cell does not divide any further. That is what you want to do for a cancer system. Now, there you would want to find which, if you put out which protein, actually all the cell cycle proteins are mutated and it does not work properly and the cell cannot divide. So predictions like this actually help in various cases. For example, drug development, targeting a particular gene or a protein, etc., etc. Right. So this is how network perturbations are viewed in biological networks. This was just an example to show how it is actually viewed. Yeah. So as I was saying, these perturbations can be used to predict diseases and understand diseases as well. So and these diseases can be termed as network perturbations as well. So cells use regulatory and signaling pathways to connect various components, which can be DNA, RNA, metabolites, etc., etc. And understanding the biological processes require knowledge of component identities and interactions. And network representation of biological systems are a powerful framework for this study of these interactions, right? Analyzing the topology and dynamics of these networks is crucial for understanding the complexity and the adaptive behavior. Now, this information with correcting two different genes or transcription factors or proteins metabolites, this has to come from some sort of wet lab evidence or some sort of evidence that is actually with conviction that can say this is going on inside the cell. This cannot be fully in silico and fully predicted. Right, there should be some sort of evidence to connect these two particular nodes or these two particular proteins and genes, etc., etc. So that information is captured from wet lab techniques mostly, and that information, if you project it in a network, you can build a whole network for all the proteins or genes in the cell and how they interact with each other. So you have a very good idea about which gene, when perturbed, can cause what disease, and on the other, in the other way around, which gene or transcription factor, when perturbed, can actually stop progression of a disease. And but one thing to remember, if you remember from the first session, I talked about biological systems being very robust. Right? So you'll have very few of these nodes that are there that if you just perturb that node, everything will be all right. Everything will go back to normal, or everything will fall apart. Because the system is designed in such a way that it doesn't allow too much importance of a particular element inside the system. Because in biology, there can be perturbations naturally. So mutations can occur naturally, which can then be selected if they are good mutations and they're doing betterment for the species, and those can be passed on to next generations as evolution, right? So there is always a scope for perturbation in biological systems, be it good or bad. So the systems are designed in such a way that small perturbations to certain elements inside the system actually doesn't perturb the system at that level. So this is how we define as this is why we say these systems are very robust in nature. But there is always a but. There can be these particular nodes or elements of the network which, when perturbed, the network can become very fragile. The system can become very fragile. And this trade-off between robustness and fragility can lead to disease formation. And certain gene mutations or combinations can trigger can cause the trigger of cascades of failures in gene-regulatory networks, leading to disease phenotypes. Examples are sickle cell anemia, which is actually, so the three examples here: Huntington's disease, which is for a repeat expansion, cystic fibrosis. I forgot the exact mutation for that, and sickle cell anemia. There's a missense mutation, right? So these are like monogenic diseases. So these are diseases which are formed if you perturb just one gene. So here, actually, though the networks are fragile in terms of these particular genes, right? If you just take a missense mutation at a particular position in the HBG, in the hemoglobin gene, it can cause sickle cell anemia, right? So that is a good example where a particular network can be very fragile if you perturb the network very slightly for that particular element. But there can be other examples as well. Yeah, there can be other examples as well. For example, cancer or diabetes or Alzheimer's diseases like these are polygenic or multigenic. So this cannot be caused by single mutations in particular genes. As the previous examples, you have to have a host set of mutations across a host set of genes to cause this particular phenotype. And not only mutations, there can be other factors involved, environmental factors, lifestyle, etc., etc. So these are polygenic diseases. So these are not formed so easily. So you can imagine these kind of conditions as where the network is very robust. These diseases are not formed if you just mutate one particular gene at one particular position. Right? So other factors, as I say, modified genes and environmental factors also play a role in disease manifestation by influencing gene interactions and pathways. So you can have both sides of the coin. So there are examples for both ends, but depending on what you want to do with the system, you can exploit the system in that way. Okay. Uh, so yeah, so I wanted to give some examples of network perturbations and uh, that are used to study diseases. So one example is basically these kind of approaches has been undertaken to investigate anomalies within cellular networks associated with different diseases. For example, a study examined the dynamic structure of human protein interaction network to predict breast cancer outcomes, right? So, what you can do, what the method followed was basically removing systematically removing nodes from the protein interaction network. Critical interaction hubs vital for the network connectivity were identified. Remember, hubs, we talked about nodes that are connected, have a lot of connectivity. So identifying those nodes are actually very important because those perturbation to those nodes can actually very easily cause the disease. So you can identify these nodes by simulating in in computation using computation, and you can schematically remove one node, a combination of nodes, and try to see what exactly the network is behaving like. Okay, again, it was identified for breast cancer, and these hubs were classified into two types. One is intermodular, and the other is intramodular, based on the correlation of co-expression with interaction partners. In intermodular hubs, crucial for network connectivity, were found to be more frequently associated with cancer phenotypes than intramodular hubs. Uh, Modularity is something that we'll discuss further down this lecture only. But just to give you an idea, intermodular hubs is basically if you consider two highly connected networks being connected by a two networks connected by a single node, or a node that is, or a set of nodes that is connecting two highly connected parts of the network. So that will be what is known as an intermodular hub. An intramodular hub will be inside that module. We'll define modules and those things later. So inside that module, how are the nodes actually connected? So two different types of hubs, and it was found that uh, cancer phenotypes were more associated with intermodular hubs, hubs that connect two different parts of the highly connected parts of the network, right? So it was found like that, and gene expression levels of these hubs were strongly correlated with their interacting partners in tumors from surviving patients, indicating the significance in cancer prognosis. And another study which compared basically miRNA regulatory networks in normal tissues and cancer tissues, and it revealed differences in hubs or highly connected microRNAs. So uh, uh, I wanted to give you an idea that you are studying all these things, actually where these things are applied. So the small parameters that we are defining, this kind of betweenness, centrality, disconnectivity index, etc., where actually is it applied? In our case, actually, insistence biology, right? So here, the example was that in normal tissues, there was a complete miRNA network, but in cancer tissues, these were disconnected subnetworks, and uh, basically some of the connections were lost in cancer tissue, and it identified crucial microRNA cliques in cancer, which are actually regulating the cancer phenotype. So yeah, understanding these become very important. Okay, I'll not go into the further examples. So I've given a couple of more examples. So one is from uh, one is from basically an example from glioblastoma cancer. This is also a cancer. So uh, it when mapped uh, cancer candidate genes onto the network, it revealed the clustering in modules enriched with known oncogenes, tumor suppressors, genes involved in signal transduction, suggesting the presence of a core network in GBM tumorigenesis. So each of this network will vary in different types of phenotypes. The connections will vary. So identifying these particular networks for particular diseases becomes very important if you want to target different nodes in the network, which means different genes or different proteins inside the network, right? Another example is for type 2 disease, type 2 diabetes, and perturbation analysis of the cellular metabolic network identified highly connected metabolites ATP and NAD+. So these are involved in different respiratory pathways and other kind of metabolic pathways. And they were found to be high contributors to type 2 diabetes. Uh, okay, so yeah, I just wanted to give you a few examples. Right, let's move on. Okay, so we were talking about modules and those kind of things, and here we will define it. So uh, one of the major algorithms for graph theory is community detection. So by intuition, what it means is, so you have a whole network. Now that network, inside the network, there will be nodes. Now all those nodes will not be equally connected or equally have the equal degrees, right? So there will be portions of the network which will be highly connected, and there will be portions of the network that will not be connected so much. So the highly connected portions of the network form what are known as communities, right? So inside the whole network, you can have n number of communities inside the network. Now, how do you define a network community? So this is actually a, a very good example to talk about network connectivity and community detection. So this was actually a thesis from Caltech from this guy called Eric Dam. So, so what is a network community? So the question is from a social network perspective. So the first question is, are you on Facebook? If so, how long have you belonged? And have you ever visualized your Facebook network? And what clusters in your Facebook do you like envision to see? Do you expect to see? Right. So in 2011, this guy was doing his PhD in Caltech, and he described and displayed a social network which he took from LinkedIn, Facebook, and Twitter. And uh, at that point of time, he had been on Facebook for 7 years, and using a visualization tool, he described the communities in his Facebook network shown in this figure below. So you have this set of, so this is the whole network, right? But this network is divided into certain hubs or certain communities inside the network, which you can clearly see by the colors of the nodes here. That is how he defined it. So uh, it quite accurately detects the communities here. The red points are your college friends. The red orange are from your college club. The green points are your high school friends, these ones, hometown folks, family, etc. And the blue points are your grad school classmates. And the peach are member uh, of a particular grad school club. The size of these nodes in the plot scale with age of the account as expected, and and his friends having the oldest account. Your college friends, sorry, the person's college friends had the oldest account, while the newest accounts were generally hometown, family, or which is given in this green color. So what it means is, loosely stated, this community detection, if you want to define the problem of community detection, it is the problem of finding natural divisions inside the network into groups of vertices called communities or clusters, such that there are as many edges within the group and fewer edges between the groups. I'll explain what this means. Right? What exactly we mean by many or few, however, is debatable, and a wide variety of different definitions have been proposed, leading to a correspondingly wide variety of different algorithms for community detection. Right? So, what it means is, so if you assume just the red dots here, these are his college friends, right? So, in it is highly probable that so these people are studied in college together, including the guy that is making the network. So it is highly probable that the connections between these particular set of nodes will be higher than the connection between the red and the green nodes, which are basically members of his older school or uh, place where he lived, etc. So inside his social network, there are there are hubs inside his network which actually denote a particular place of origin. Right. So how you define a community? You define a community by saying the set of nodes, say for example this red set of nodes, which have high number of connections within each other and less number of connections between that community and the other communities. So this is a community because there are high number of connections within this particular set of nodes, but there is very less number of connections between this whatever yellow colored set of nodes and the blue color set of nodes. This is how you loosely define a community. Now, mathematically, this becomes much more complex to say, like, how will you define, like, what is the optimal number of connections within that particular community to say that's a community, and it is, I mean, how many should the how exactly how many edges should be present inside a particular community compared to between communities. That is something that you need to define mathematically. That becomes a little bit more complicated, but intuitively, if you try to understand, it's basically you want to find places in the network which have very high connections among themselves but very low connections between themselves and other parts of the network. So that is how communities are defined inside a network. Now there are various algorithms to kind of detect communities inside the network. We'll talk about one of them, which is the Gerber-Newman algorithm in the later slides. But this is what community detection actually, the problem of community detection actually means. Now, if we talk about biological networks, how will these communities look like inside biological networks? Inside biological networks, these communities will be set of proteins or set of genes which have similar type of function or which are dependent on each other for functions. Say, for example, if you plot the whole metabolic network here, right? You have pathways like Krebs cycle, glycolysis. You'll have other pathways, for example, right? So if you imagine just these two pathways, Krebs cycle and glycolysis, there are elements of there are molecules of glycolysis that is used up by Krebs cycle, but glycolysis itself will be much more connected compared to connection between glycolysis and Krebs cycle. So glycolysis and Krebs cycle will be individual communities inside the network, right? So uh, the concept of communities is defined like that. Now, why would you want to find communities? Because you would want to find these connected components inside the network and study why these are so connected. If these are so connected, that might be there not might be, there will be a biological significance to it. Either they are part of some pathway, or they are working together, or something like that. But there will be a biological explanation for that. So that is why you want to detect these communities, and this is one of the very famous problems from graph theory, is which is known as this community detection problem. Right? I hope I have given you the intuition behind the problem. Right. Now, yeah. So these are some examples. So where we use some examples of community detection. So a promising computational approach to discovery of functions of genes and proteins is to identify functional modules inside biological networks. Since modules or communities are sets of genes or proteins that perform biological processes together, as I was saying, glycolysis and peptidal, something like that, it is possible to classify proteins with unknown function by determining what community they belong to. Right? So maybe you have an unknown protein, you do not know the function of that protein, but you know that that particular protein belongs to a hub or a community inside the network, which is responsible for, say, for example, a particular molecule's uh, metabolism. So it is highly likely that that unknown protein is involved in that particular function as well. Right? So correct identification of functional community has also become an important biotechnological and drug design applications. In many cases, the deletion of a certain function may be necessary, and this can be achieved by removing the entire functional module. So in general, as I was saying, the biological networks are very robust. If you perturb a single gene, it is highly unlikely that you will be able to remove that particular phenotype that you want to remove. So instead of targeting a single gene, you target a whole community, a set of genes, a set of nodes. And if you target that whole set of community, it is highly likely that you can have your desired result. Uh, Targeting single genes or single proteins is not the way to go. So here comes another caveat of systems biology, is this is the importance of looking at a system from looking at a biological cell or a biological phenomena from a systems perspective. So earlier, what used to happen is you knock out a particular gene in using molecular biology techniques, and then you want to study like how the phenotype is affected. If you are able to get past the phenotype or something like that. But that approach is not always correct because your biological network is very robust. If you just knock out that particular gene, it is highly likely that there's some other gene, other protein will be able to kind of mimic its function, mimic its expression, or like bypass its function to do something else. So it's highly important to see this as as a network, as a system. The moment you're seeing it as a network, you're seeing it as a whole system, and then you can identify these particular hubs or particular communities inside the network you know are responsible for this particular function, and you want to remove or target this particular community, not a single gene or a single protein or a single transcription factor. Right? And communities also play a particularly important role in understanding human diseases. And proteins that are involved in the same disease, obviously, as I was saying, they tend to interact with each other, so they will be part of the same community. Uh, in the disease gene network, in each node is a gene with two genes being connected if they are implicated in the same disorder, and the size of each node is proportional to the number of disorders in which the gene is implicated. So this is basically the human disease network and the disease gene network. So if you see, say for example, for different disorders, there are the genes actually cluster together. So for bone disorder, you have this set of genes that are working together. For connective tissue disorders, you have this set of genes that are working together. So identifying this particular set of genes, this communities inside the network is very important to understand uh, w- what particular nodes to target, what particular genes and proteins to target, and so on. Okay, moving on. Now we'll come to the algorithms. Sorry. Yeah. Okay. So one simple measure is to use a hierarchical clustering method, which is basically you cluster the nodes that are at a certain distance, within a certain distance to each other, and you go on doing it iteratively till you find a network which has no more closest genes to each other. So inside the network you find a network which is the community of the network, right? So carrying out hierarchical clustering requires two decisions, which is one, first is the distance metric. How are you going to define a distance between two nodes and measure the distance, and the decision of how to extend the distance function to define the distance between any two clusters of node. So, so you start with one node, you see which is the closest node to that, you take that inside this cluster. Then you have a set of two node cluster, and you see which is the third node which is closest to this particular two node cluster. Then you take that node inside that cluster. Like that, you go on hierarchically building a community inside the network. And so you have to define two things for that. One is the distance function actually, and second thing is basically the if the minimum distance between two such communities. What and how it should be. Right. So these are certain distance functions that is used. So this is the Euclidean distance, which is the most straightforward. It's just square root squared of the coordinates between them. But there are certain differences in this distance function. One is a cosine similarity function. This is, if you remember, if people are from the mathematics background, or if you remember your mathematics, this is the basically the dot product function. Right. So you have different functions to define. Simply, you can define Euclidean distance or cosine distance, or you can define Pearson correlation coefficient, which can be used as a proxy for a distance, or you can do a lot of things. Here you have that mathematical uh, freedom to define what you want to define. Now, why these distances are important? Say, for example, what is the difference between a Euclidean distance and say for example a distance metric like an entropy, right? Entropy can also be defined as a distance, right? uh, you'll notice that this Euclidean distance is actually, there's certain properties to these distances, right? Euclidean distance is actually symmetric. The distance between from point A to B is the same as the distance between point B to A. Now you imagine you are in a directed network. That this property actually does not hold. So if you are in a directed network, the distance between A to B will not always be equal to distance between B to A, depending on how the nodes are connected. So how are you defining a distance becomes very important. Are you and what type of network you are working on? That is what will prompt you to define the distance metric. So you can, in those cases, you can use something like these. Say, for example, you can use the covariance Pearson correlation metric, which can be which is basically non-symmetric. You can use a symmetric if you have a non-directed graph. A symmetric distance metric would do. In which case, you can use Euclidean distance and so on. Okay. Okay. Now we come to the algorithm, which is the Kerr-Newman algorithm, uh, uh, which is basically a refined version of the hierarchical clustering method. The idea is same, that you have a distance uh, and you kind of cluster nodes which are closely related to each other. So the idea is same, but here is the algorithm. So at beginning of each iteration, you compute the betweenness of the edges in the graph and remove the edges with the largest betweenness. Now imagine what the betweenness means. So betweenness is defined by a betweenness of a node is defined by how much that node is in the middle of this of the shortest distance between two other nodes. So in the first case, say for example, this is the network. Forget about the circles on top of the network. This is the network. Which node has the highest betweenness here? In this case, this will be E, because this node actually connects a shortest distance between a lot of nodes. Say F and A, the shortest distance is this, E connects that. H and B, the shortest distance is this, E connects that. C and G and C, the shortest distance is this, and E connects that. So E the is the node with the highest betweenness centrality. Why you are looking for highest betweenness centrality? Because you want to find the nodes that are actually in between these communities, this set of clusters of nodes. You want to find the nodes which are that are connecting this set of clusters. So that is why you want to use the betweenness centrality metric. Right? So if you have in the first iteration, what you do is calculate this betweenness centrality metric for each of the node. And in this case, if you can calculate for all these A to, I think, I, the maximum betweenness will be for E. Now, the idea is you remove node with the highest betweenness. So at the beginning of each iteration, you compute the betweenness edges in the graph and remove the edge with the largest betweenness. And if more than one edge has the largest betweenness, you remove all such competing edges at the same time. If the graph gets disconnected to two or more communities, we compute the total modularity score of the resting communities. I'll come to the modularity definition in the next slide. And you repeat the iterations until there are no more edges. And the partition with the total modularity score is the optimal partition. And you could stop dividing a community if the total modularity score of the undivided community is greater than the total modularity score of the divided community. So in this case, edges with the highest betweenness centrality will be FE and EA, which basically is the E node is the node with the highest betweenness centrality. And if you remove these two edges, the next set of nodes will be, now this connection is not F to E and E to A is not there. Now you have these two set of communities inside. Now if you compute betweenness values of all these four nodes, this will be exactly same. So there is no point in dividing up this particular community. It's the same in this case. So you can stop that iteration there, and you can claim that these are the two communities inside the inside the network. Okay. So I was talking about modularity. Now, how do we define modularity? So the community structure problem differs crucially from graph partitioning problem in that you do not know what will be the final sizes of the community that you are expecting, right? So if the community sizes are unconstrained, then we are, for instance, at liberty to select a trivial division of the network that puts all the vertices in one or two groups and none in other, which guarantees we will have zero intergroup edges. This division, in a sense, optimal, but doesn't really tell us anything of what, right? And the idea that the true community structure in the network corresponds to a statistically surprising arrangement of edges can be quantified by the measure of something known as modularity. And the modularity is a multiplicative constant, the number of edges failing within the groups minus the number of uh, expected edges inside and in an equivalent network with edges placed at random. So what it is saying is it is how you define modularity is the number of edges that fall inside one of the groups. So these two groups you have divided up. You'll calculate modularity on these two groups. So the number of edges uh, falling inside these groups minus the number of expected edges in an equivalent network with the same number of nodes and with the edges placed at random. So what you're saying is statistically, if you have another network with the same set of edges and a same set of nodes and edges placed at random, does your divided community have a better modularity compared to your uh, previous community? This is what you calculate, right? And this measures what it measures is actually the strength of the community partition by taking into account the degree distribution. And now, given a network with m edges, we did a problem like this in the last class as well. So give a network with given a network with m edges, the expected number of edges between two nodes I and j with degrees di and dj are respectively the multiplication of di and dj divided by 2m, right? So expected number of edges between, say, for example, node 1 and 2 is going to be 3 into 2 by 2 into 15. So there are total 15 edges here. Total, if you count, there will be 15 edges, which is the value of m. And the between 1 and 2, uh, you have to find the degrees of 1 and 2. What is the degree of 1? The degree of 1 is 3 and the degree of 2 is 2, considering you have already considered this connection. 1 and 2 connection you have considered. So the expected number of edges will be given as 3 into 2 divided by 2 into 15, which is zero point 0.2. Right? This is how you calculate the modularity. So, and this is, and you sum that up over all set of edges, all set of nodes, and you calculate the modularity of the total partition of the total network community. And if the modularity of the network community is more than the modularity before, then that net community actually holds, or else you go back to the previous partition, right? So a larger value for Q indicates a good community structure. Okay. This is how you define that modularity. Okay. Now, uh, the idea behind Garvan-Newman algorithm is that you maximize modularity. This is known as the modularity maximization. So the intuition behind the idea of modularity that a community is a structural element of the network that has been formed in a manner that is not a random process. So again, the idea of community comes from that you have these connected hubs inside the network, but these are not at random. There are biological reasons. In our case, biological. In other cases, there can be other, like whatever nodes, whatever network you're considering. There can be other um, reasons as well. But in our case, if we consider biological networks, there has to be a biological reason why that particular set of nodes are actually clustered together. So the idea is that clustering has happened in a non-random manner. It's not a random process. So there is a certain reason behind that clustering. And if you consider the actual density of the links in a community, it should significantly be larger than the density you would expect if the links in the network were formed by random processes. That is the idea of modularity, right? So you basically, this part of the equation is telling you what is the number of expected edges that can be formed if it was formed at random. Right? And so, yeah, so for example, if you have two nodes I and J, they end up being in the same community. There should be more than a link between Aij. Sorry, there should be more likely a link between these two nodes, and leading to that is basically Aij will be equal to 1, leading to an overall higher value of for modularity. And if I and J, the two nodes, being in a community such that the chances of having a link between them is just same as any two nodes in the network, that is in a random case, then the value of q is more likely to be low, because there could be some in some random processes, in some cases there might not be connections between I and j. So what it is telling is, if there is a connection between I and j, and they're in the same community, there is a reason behind that connection. Because in if you do it randomly, if you just take these two nodes and inside the network and try to randomly put in edges, there will be a lot of number of cases where I and j will not be connected. So if that is connected, it is highly likely that they are connected for a reason, and the value of q will be more compared to the value of q if there is no connection between these two particular set of nodes. Right. So I have talked about a lot of things. Let's take an example to understand how this actually works. So this, you have a network. Uh, you have different nodes, and these are connected like this. Now you want to implement an algorithm which will partition this particular uh, network into whatever number of communities that is present actually present in the network. Right? Okay, so you calculate modularity between all set of nodes. So one, uh, uh, so basically you have 7 nodes, 1, 2, 3, 4, 5, 6, 7 in on the left and right. So for the Garber-Newman algorithm, you calculate the betweenness centrality for all the nodes first, and then you see that this particular edge, if you calculate the betweenness centrality, you'll find that this particular edge has the highest betweenness centrality. So in the first step, going back to the Garber-Newman algorithm, in the first step, you calculate the betweenness of all the edges in the graph, and you remove the edge with the largest betweenness. So in this case, the largest betweenness will be between 7 and 8. So you remove this particular node. Now you are left with these two set of nodes as two set of communities. Now you calculate modularity inside these particular two communities and see if what is actually the modularity score. So you, on the left you have nodes 1 to 7, and you calculate the modularity. So modularity is a pairwise index, so you calculate for each pair. So 1 and 2, 1 and 3, 1 4, 1 5, 1 6, 1 7, right? And then you go for 2 again, 2 3, 2 4, 2 5, 2 6, 2 7, and you do all these possible combinations, and you get values for each of the pairwise modularities, and then you add them up, right? So if you do it for 1 to 7 on the left hand side node, it's 4.385, and on the right hand side nodes, if you go from 8 to 17, the modularity is actually the same, because this is a symmetric network, it will be the same. So the total modularity score is 8.77. This is your first iteration. You have broken down this particular edge, and then you have calculated modularity of these individual clusters, and then you have added them up, which comes to 8.77. Now, if you do not know if you want to stop here, there might be other communities inside these two communities. Now, how will you know that? In the second iteration, you go with the Garber-Newman algorithm. You'll remove the edge with the highest betweenness. Right. If you remove the edge with the highest betweenness, this will be these two edges. So if you just imagine these two network, so this edge connecting 7 has the most like number of shortest paths in between. passing through 7, right? So these two edges, this 12, 12, these two edges are actually the uh, the the most betweenness edge of this particular network, right? So you remove in the next iteration, you remove these two edges now. Now you are left with if you do it for both the communities, you are left with structures like this. So you have 1, 2, 3 connected, 4, 5, 6 connected, 7 as a separate one, and 8 as a separate one, and then you have 9, 10, 11 connected, 12, 13, 14 connected. Uh, uh, you have uh, you basically have these set of nodes that are connected, and these connections are removed now. 8, 9, 8, 12 are removed, 3, 7, and 6, 7 are removed. Now, if you calculate modularity between 1, 2, and 3, this network, you calculate the modularity by that formula. You compute modularity for all the combinations. So 1, 2, 1, 3, 2, 3, and there is this connected component is also there, which is 1, 2, 3. Right? Sorry. Yeah. This connected component is actually also there, which is 1, 2, 3. Now you calculate for all. So 1, 2, 1, 3, 2, 3, and 1, 2, 3. Similarly, for more, and if you add these all up, it's basically 2.53. Right. So for modularity, same for modularity 4, 5, 6, you calculate uh, 4, 5, 5, 4, 6, 5, 6 like this, and you calculate, you get this, since this is symmetric, you'll get the same value. So yeah, for all of this, you get 2.53, 2.53, 2.53. Now you have four of these, right? These will have zero. You do not consider this, and you add all this up. So the total modularity score becomes 10.12. Right. So this modularity score, 10.12, is greater than the previous modularity score, which was 8.77. So what it means is this is a much better formation of clusters than the previous set of clusters as we defined here. So if you what you want to do is maximize the modularity of the network, right? So more the value, the better. So if you have more value, you accept the next set of partitions and reject the older set of partitions. That is what you do here. You went from 8.77 modularity score to a modularity score of 10.12. That means this is a better partition than the older one, which is this. So actually, the final partition. Now, if you remove further edges, what will happen is the modularity score will decrease because these connected components will be gone, and the modularity score will decrease. You can try doing it, but it will decrease. This is the maximum modularity score that you'll ever get, which is 10.12. So this is actually the best partition. So how many clusters have there been found? 1, 2, 3, 4, 5, 6. Right. So this was an example for a simple network. This is a quite simple network and a symmetric network. Uh, and the idea is for Garvan-Newman algorithm is that you find the edge with the highest betweenness. You remove that edge slash edges if there are multiple edges with the same betweenness value, highest betweenness value, and then you calculate modularity inside this network and you add them up, right? So uh, the maximum modularity configuration will give you the best partition. The idea is that. Okay, so I hope I have been able to make you understand what actually this modularity maximization and Garvan-Newman algorithm actually does. So this is an example. You can try it with like other examples. There will be a lot of examples on the internet. You can try it with other examples and see. But this actually works very nice. But there are shortcomings of these two. It cannot be used in very highly complicated network and so on. This breaks down when the network size is very complicated. But this is a very good algorithm to do in a decently sized network. And the final thing that
Soma,
yeah.
uh, what does this high modularity indicate in terms of biological networks? Does that mean that the different modules we got after doing this whole exercise? they are independent of each other, they are functional on their own, or what does it?
Yeah, you can say it like that, but technically, what it means is basically these modules are more connected with each other. From from a graph theory perspective, these modules are more connected with each other than with other modules or other edge, nodes in the network. Now, what this connection actually means can differ in different conditions. If in a protein-protein interaction network, you can say that these proteins actually work together. They have a similar functional consequence, so these have high modularity. But in, say, for example, in other cases, say these three elements are, one is a disease, one is a gene, one is another transcription factor. In that case, what it means is basically the higher modularity for that particular network, for that particular community of node, means that those particular nodes are involved in that particular disease. So the meaning will differ from network to network. Whatever network you're trying to study, what are the elements of the network, the meaning will differ. But what it intuitively means is that these set of networks actually cluster together. They are more connected to each other. They form a community stronger than they form with other nodes in the network. Now, how you interpret that will depend on the network that you are studying, right?
Okay,
Yeah.
thank you.
Okay. So, yeah, let's move on. So, this is the last part of today's session, and which is basically the concept of network motifs. Uh, Now, what you're finding here are actually like when you're doing this network divisions or net community detection inside the network, what you're finding is basically subgraphs inside the network. So these are actually subgraphs inside the network, which which are present, which have some which will have some significance depending on the network you're using, as you asked. Now, all of these subgraphs will not occur with equal frequencies. So this particular concentrated network of nodes will not occur with equal frequencies. Now, uh, some of these uh, some of these communities or some of these set of clusters of nodes will be overrepresented compared to a randomized version of the same network. So if you say, for example, you have 10 nodes and you're randomizing the connections between all these 10 nodes, and you compare all these random connections and of the particular nodes forming a network with your original network, and then you see inside your original network, you have an overrepresented set of these subgraphs, which are not actually overrepresented when it is coming randomly. What it means is that particular section of the graph or that particular subgraph is not there by random. There is a biological significance to it, right? That, as I was saying in the other cases as well. So these are what are known as network motifs. So network motifs are subgraphs inside the network, which are occurring at a frequency more than compared to a regular randomized version of the same network with the same number of edges with the same number of nodes. Right. So to identify these motifs, you have to identify subgraphs of n nodes in the network. Then you randomize the network while keeping the number of nodes, edges, and the distribution unchanged. And I identify these all subgraphs of n nodes or in the randomized version. Subgraphs that occur significantly more in the real network compared to a randomized one are designated as network motifs. Right. So there are models to identify network motifs. So for example, you have configuration model, which basically generates a random graph with an arbitrary sequence of degrees. So for example, you have uh, s- uh, this set of degrees, arbitrary sequence of degrees for 7 nodes in a graph. So these are no degrees for each of the nodes. So node 1 has degree 3, node 2 has degree 4, and so on. So these are for 7 nodes in a graph. Now node I can be given ki stubs or of edges or half edges. So what are stubs? Basically, you imagine two nodes connected, and if you cut the edge from the middle, what remains is half of the edge and the node. So half of the edge is known as a stub. So when you're cutting these things, these edges, what you're remaining is are stubs, what are known as stubs, which are known as half edges, basically. So randomly a pair of randomly you sample a pair of unconnected stubs and connect them. So you have this set of stubs. So for example, node 1 has 3 stubs because it will have 3 edges. So 3 stubs, 4 stubs for node 2, and so on, 5 stubs for node 3, and whatever the degree distribution for each of the nodes is. Now you have these nodes with the stubs, and you randomly take these nodes and you join these stubs, right? So what you're doing is essentially keeping the degree of each of the nodes same, the number of the nodes same, and you're just reconnecting the network. So what you're generating is a random network, and you repeat till no more stubs remain, and you generate a random network. And now you compare this particular random network to your own network and see if there are certain subgraphs inside the network that are occurring more frequently in your network compared to this randomized version of this network. So there will be not one version of this network, this randomized version. You can have like n number of versions of these networks depending on how you connect these particular nodes and edges. So across, say for example, you generate 10 of these random networks, and in all, among all the 10, you see only in two of those networks there is a particular subgraph that is present, but inside your graph that is present quite strongly. So or two or one of those networks, so 1 in 10 times in a random network that subgraph is present, but in your original network that subgraph is present. So that means it is very unlikely that particular subgraph is present due to a random process, that is present actually due to some sort of biological significance or whatever significance you are studying, right? So these two models, a configuration model and edge switching model, are used to generate these random graphs to be compared against your original graph. So the configuration model works like this: you just take the nodes and the edges and you cut them off the middle, and you are remained with that node and stub, which is basically half the edge. And in edge switching model, you select a random pair of edges and you just rewire them. You take that edge and you move that edge somewhere else. And you repeat that rewiring of the network and you produce a randomized version of the original network. And so repeating this set of like this process for n number of times will give you n number of these randomized networks. Now in n, say for example, n is very high, n is 10,000, and in 10,000 across 10,000 of this network, you find in one network only you are having that particular subgraph that is present in your particular network, which means that that particular subgraph is actually a network motif. It is more, it is statistically significantly present in your graph compared to 10,000 of random networks. So this is how you find network motifs. So the idea behind finding network motifs is you have the same set of nodes, you have the same set of edges. If you rewire them somehow, and and you rewire them somehow to get a randomized version of the same graph, and you do it a huge number of times, and among them, only in a very few number of times you find the kind of subgraph that you have in your network. Most likely, that particular subgraph in your network is actually a network motif. Right? So that is how the concept of network motif goes. So yeah, so that's all from that side. So we'll move on to the questions. Before that, if anyone has anything to ask. Okay, then let's move on. Yeah. Okay. 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 So the answer is both one and two. So first one is simple, like a fixed number of edges you have and a fixed number of nodes you have, and you're randomly connecting. That is by definition a random network. Now a pair of nodes, n number of nodes is fixed, is connected if a number x generated randomly exceeds a fixed probability, and if x is less than p, then the pair of nodes are not connected. Yeah, this can be another strategy to generate a random network. But the thing is here, the caveat will be that the number of edges might not be the same as the number of edges in your original network, right? So the only caveat is this, but that is not a bottleneck for this. Only if you're doing some sort of analysis of, say, for example, you want to find a network motif or something, then you need the exact same number of edges and the nodes. Other than that, if you just want to generate a random graph, it's possible to generate it. In some cases you might not need the same set of edges. You just need the same set of nodes, and you want to find, compared to a random network, is the number of edges much more in your network compared to a random network. In that scenario, you would use a strategy like the second one. So the answer is you can have both the strategies to generate a random graph, right? Depending on your need. If you are doing a network motif, most probably you will not use the second one, you will use the first one. But if you are using, want to generate any random graph, both works. No, not a problem. Okay, answers. I'll give you another minute. So I think this question will relate to ER networks more. I think I'm not sure if this is covered in this class, this week or the next week. Maybe I'll cover it next week, but I'm not sure if Sir is covered in this particular week or not. But this question relates to the ER random graph, which is the Ardor Shreni. I think this will be covered in the subsequent classes. So the idea behind this particular question is basically when you have an ER graph, uh, the edges are placed at random between two particular nodes. If you're generating an ER random graph, you have a fixed set of nodes, and you're placing edges between this set of nodes and at a certain probability. So yeah, keep that information while answering this. Okay, I'll move on because we are running a little late. So, yeah. So the answer is true, and the explanation is that the degree of a node gives node information about the number of connections amongst its neighbors, right, in a random network. And local clustering coefficient of a node is a measure of the number of connection amongst the networks of the amongst the neighbors of the node. It's not about the degree of that particular node. The the clustering coefficient depends on the neighbors number of degrees around the that particular node. So the clustering coefficient will not depend on the degree of that particular node. It will depend on the degree of the nodes around itself. So the answer is actually true. So if you do a local clustering coefficient of a node, it will be independent of its degree, and this is very much related to the ER networks that are there, random networks, Ardos Sreni. I have not covered it, or like maybe next class I can cover it. You can just look it up. It's basically, I'll just show you here. Yeah. Uh, so yeah, so this is the model G of n comma m, where basically m is the edges, n is the number of nodes. The nodes are considered to be labeled, meaning the graphs premium permitting also. Yeah. So this is a better definition. So in this particular model, a graph is constructed by connecting labeled nodes randomly. So there will be a probability associated with it. This is the probability which is calculated, and according to that probability, each edge in the graph will be connected between two particular nodes with that particular probability, independent of every other edge. So the degree distribution actually doesn't matter for that particular node, right? So I think we'll cover this in a little more detail in the next classes. Let me see. I haven't checked uh, the things that have been covered in the next class. So I'll look into it and I'll maybe cover the ER networks a little bit more. No, it's actually, if you go back to the dissortativity definition. Yeah. So dissortativity is given by this formula, n naught minus nv by n naught. So nv is the number of vertex pairs that remain connected following the removal of vertex v. So higher is your dissortativity, the less resilient is your network. So the lower dissortativity means, lower disconnectivity, sorry, not dissortativity. Lower disconnectivity means more resilient is your network. So if your n minus v is 0, n naught by n naught becomes 1, which means like if n minus b is 0 implies that if you remove a particular node, the whole network collapses. So it's a form of least resilience or robustness. If you go back to the question, so if it is disortative, it means the higher value of disortativeness or disortativity, the higher the value, the less resilient it is. So it is actually saying the opposite. So disortativity results in resilience to random perturbations but is vulnerable when hub nodes are targeted during perturbation. Okay. So higher your value, the less resilient it is. Higher your value, it means like you remove that particular node, the the whole network collapses. That is what a higher value of dissociativity or disortativeness means. Right? So it is a false statement. These
But
are
doesn't it mean that when disortativity is high, then this question says that we are randomly removing a node, which
yeah
doesn't necessarily mean that we are targeting a perturbation in a hub. So that removal can definitely be a node which is not really well connected to the hub or doesn't have many connections. And it
yeah
is not very central, so it will not have any effect on the.
I understand what you're saying, but the point is if your disortative index is higher, if you have a higher disortative index, what it means is you have a lower resilience. So if your network is disortative in nature, it generally has a lower resilience. And if you just forget about the hub explanation for a moment, if you just randomly remove a node from the network, it means that your network is going to like fall apart very easily. That is what disortative means. Like higher value of that index will give you a lower resilient uh, network. So I think there is a uh,
So
I think
actually,
this
what I read about this concept of assortativity and disassortativity is that in assortativity, hubs are connected to hubs, like same type of things are connected to same type of things. So
yeah
more nodes are connected to each other,
yeah
more hubs are connected to each other. And the opposite is true for disassortativity. So if there are more hubs which are connected together, and
hmm.
if you randomly remove something, then the other hubs will also be affected equally. And that will make the network more fragile, isn't it? But if there are more independent nodes and less hubs, and if you randomly remove a node, which will be more likely because you're not targeting any specific perturbation to a hub. So in that case, that network will be more resilient.
Yeah, that holds. Basically, yeah. So how assortativity is defined is like, yeah, as you said, ki like same similar type of nodes connected to each other. But what happens is if uh, just let me get my thoughts for a moment. So you have similar type of nodes connected together. Now you randomly remove a node. Uh, that hub is going to be more affected than randomly removing a node from a disconnected hub. That is what you are saying, right?
Yeah.
Yeah, that is true. Mm, okay. Yeah, that is true. Okay, I'll look into this market here. The thing is, these
Yeah.
answers are
Yeah, they are very tricky.
no.
Even the clustering coefficient thing uh, like indirectly depends on the number of nodes, the node which we are finding the clustering coefficient for is the degree defines how many neighbors it has.
But the
So
clustering coefficient itself will not depend on the degree of that particular. So it will depend on
yeah,
the there is a slight
so it
caveat to
was
that.
confusing that whether it is dependent or not. So because
Yeah,
it
but
defines the number of neighbors and then
but
you calculate
yeah,
clustering
yeah, I
coefficient.
get it. But
Yeah,
in general, the if when you're calculating the clustering coefficient, you do not take into account the degree of that particular node. You see how well
yeah,
your neighbors are connected. But
yeah,
technically,
true.
the higher the degree of the node, the more neighbors it will have. But yeah, it's the questions are a little. I understand. Okay, I'll look into this.
Thank you.
Yep. NPTEL has some weird questions. I'll be back in a second.
Hey, we can't hear you.
Hello, Soma, we can't hear you.
Can you hear me now?
Yes.
Yeah. Okay. So, yeah, in a random network, there will be no special nodes. Obviously, you are just interested in the number of nodes. Any two nodes will equally likely to be connected. The equal likeliness will not be half or one or whatever. It will be depending on the distribution from which you are picking. It might be like a one third or something like that. But the probability of two nodes connected will be equal unless and until you define a probability function which actually gives you random probabilities, right? Like in a ER network. But other than that, the probability will be equally for all two nodes to be connected. And if you have a degree distribution, if you have p equal to 1, what it means that all nodes will be equally connected. All connections will be there. Like exhaustive number of edges for all the nodes will be there. So the degree distribution will be uniform. So yeah, I hope this is clear. You can see the answer here. So yeah, A and B and D, all three will be there. And there are no special nodes because you're just interested in the number of nodes, like not any special kind of nodes or anything. Yeah, the other three are like weirdly false. So yeah, so it has a functional significance. Motifs will have certain sort of some sort of functional significance depending on the network you're looking at. Yeah, and it can be found in all other kind of networks, not limited to biological networks, and it can be found in all types of networks, smaller, bigger, doesn't matter. A similar type of concept for motifs is actually in this motif detection. I forgot to mention actually one example, very good example of motif detection is actually during genome assembly and detection of transcriptionally regulatory motifs. So these motifs will be inside the genome where these transcription factors will go and bind. Now these motifs will have very these set of sequences which will be very similar to each other across the genome, and these transcription factors will come and bind specifically. So for example, one transcription factor can regulate say 10 genes. Now that transcription factor will go and bind to the promoter of those 10 genes, and the sequence will be very similar of those 10 genes promoters where the transcription factor will go and bind. So identification of these motifs inside these genomic regions is also a very good example of an application to this. Yeah. Okay, the question is that a directed protein-protein network has been constructed, and we want to identify protein clusters that interact densely with each other but have sparse connections to other proteins. This is the definition of a community, right? Which method would be most effective? Uh, Basically finding subgraphs that occur significantly more in the network when compared to a randomized network. So network motives or net community inside a network, this is how it is defined. So you have a set of nodes inside the network which are highly connected to each other and very less connected to other nodes of the network. So that is how it is defined. Uh, this is also a weird, tricky question. Yeah, this can have multiple answers. Okay, so technically the given answer is zero. I'll tell you why it is zero. So they're considering like there are five nodes in the network and everything is connected. So the whole graph is connected. So there are no individual subgraph containing most connections. So from that perspective, it is zero. But if you want to say, for example, you want to find motifs of only three uh, nodes inside the network, so then it will be like 5C3. If you want to find motifs of like say four subgraphs inside the subgraphs containing four nodes inside the network, that will be like 5C4. It can go on till k equal to 2. The thing is, here the logic that that is being used is your whole graph is connected. There is no concept of a motive in a fully connected graph. The concept of motive only comes when there is like a subgraph inside the graph which is more connected than the rest of the graph. That is the concept of a motive. So from that concept, the answer will be zero. But if you want to find, you can say the whole graph is a motive. But technically, the definition of a motif is like it should be present. It's a subgraph inside a graph. So from that perspective, it's zero. But if you want to say, for example, find motifs of three nodes inside this particular five node graph, so it'll just be 5C3 like that. So you can do a lot of combinations, but the concept is that a motif should be a subgraph inside a graph, which will be more connected than the rest of the graph. So from that concept, it's zero. So I'll not dwell on this much because the questions are a little bit. I get it. NPTEL has a little weird questions. So, okay, so the highlight here is basically complete. The graph is complete. The if in a complete graph you cannot have motifs inside. Mm, yeah, I'm not sure about this as well. Um, Yeah. So this is actually a directed graph. If you consider that, then the option C holds. So you have connection from 1 to 3, 3 to 5, and 5 to 1. So that forms a strong connected component. Right? 1 to 3 and 5. 1 to 2 there is no connection. There's 2 to 1 connection. Right? So the first option is not there. It's not symmetric because 1 to 2 and 2 to 1 there is no connection between 2 to 1 to 2, right? There is a connection from 2 to 1. And from 4, you cannot move to 5, you cannot move to 1. So there are no uh, existing paths between node 4 and every other node in the network. 1, 3, 5 is strongly connected because 1 to 3 is connected, 3 to 5 is connected, and 5 to 1 is again connected. So that is a strongly connected component. You have to consider this as a directed graph. This is not an undirected graph. If you consider this as an undirected graph, this is a fully connected graph. So this is not an undirected graph. This is a directed one. And yeah, option C is the correct one. Any questions in this? All right, moving on. Okay, NetworkX is a Python module, Python package. Um, but you will mostly use MATLAB, but also NetworkX is you can be a little bit familiar with NetworkX. It's a very useful module. So yeah, this is how a graph is defined: G equal to nx dot graph. Nx is the NetworkX module, and this is how you add edges. This is how you remove edges, and yeah, it's quite simple. Yeah, sorry, this is how you add edges and remove node in this case. This is a simple one. Yeah, the answer is two because I hope the answer is two. Yeah, the answer is two. Yeah, because you remove the second node, only 1, 3 and 1, 4 remains. The rest of the connections go off. So there's only two connections that remain. Okay, that was all from my side. I'll look into this question. I'm not fully certain of this question. This is a weird one. I'll look into it. I, I've marked it. Hope I don't forget. Uh, okay, then if you have any questions, you can ask, or else we'll close this session.
I have a question.
Yeah.
So there is a question in the assignment for this week,
Okay.
in which we are given a CSV file, and the header says that it's a matrix, and the header just mentions the coordinates are symmetric, but the coordinates which are given, like suppose 344, node 344 is connected to node 1, that is given. 344 and 1 is given, but
Yeah.
1 and 344, this set is not given. So,
Uh,
and then we are asked to,
I am not sure. If you can show me the file, I can maybe comment on that. I have not seen it. I do not have access to those. So if you can show me, share your screen, show me, I can maybe comment on that.
okay. I'll try to look.
Yeah. So it can be possible that they have given only one side of the connections and the other side you're told to assume that it is a symmetric network. You have to assume that the other side is also there, something like that.
Can you see in the chat?
Okay.
So there's more data, but in this format.
Yeah. So what they're saying is like they have given like the connection between 344 and 1. Uh,
Yeah, but they have not given the inverse connection.
Inverse connection, you have
So
to assume that it's symmetric. That
yeah.
is what is.
So in that case, before calculating anything, we should transpose this, right? To because this data will represent half the triangle of the
Yeah,
matrix.
yeah. This will basically be the top half or the bottom half of the matrix. You need to get
Yeah,
the rest of the, yeah. So basically, you transpose this and you add these two things. Not add is in plus, just put
yeah.
these two together. You'll have the whole matrix. Yeah.
Okay, okay.
Yeah, actually, I'm not
Um,
familiar. Basically, I don't have access to these things because I myself am not registered to this, so I do not get access to these things. I
okay,
only have access to the discussion forum and discussion forum also I have access to, I think, last year's. I only have this thing, the session only. You can, if you have
okay.
any issues regarding assignments or anything, you can post on the discussion forum. Somebody in the from the course module team will actually reply back to you, or you can mail them.
Yeah, okay.
Okay, thank you.
Yeah. Thank you.